we got all three of us to do this today. Greetings from North Carolina State and Karuba from Missouri. Yeah, we have never met each other except by Zoom. So putting this project together and spray drying on two ends of the country. So this is, it was actually the different title. This is tomorrow's workshop talk too. We're talking about novel added ingredients from elderberry, phytochemicals and things, more or less. We need a slide. Okay, Karuba. All right, I introduce us. All right, Karuba Krishnaswamy, thank you. I didn't have time to practice. And then Roberta Hoskins, who is from Brazil originally, and is now working at NC State with us. Marvin Moncada, who joined our team recently from Louisiana State. He's in food, these guys are in food science, by the way. I'm the token horticulturist. And then Marianne Lytle, who's also our center director. Okay.
Yeah. Part of the research was done at Mizzou, and part of the research is going to be conducted at NC State.
Okay. So this is where we deviated because I was going to put this plug in for tomorrow, but basically a lot of people don't know who the heck we are. We're the North Carolina Research Campus. We're not in Raleigh. We're actually near Charlotte, which is at the south end of the state. We used to be a textile mill. We're the biggest textile mill in the country. It was imploded in 2002. And this whole research institute was built by a billionaire. And the idea was to study health. And the idea is to bring together a number of universities. And the plants were, all right, here, hold this. Sorry. I guess this is the, yeah, there we go. We're practicing for tomorrow. So anyway, there, okay. Focus on health and plant-based diets. The idea was we don't want to do animal work in terms of meat or dairy. It's about plants at the university. Multiple universities came together. It's a fairly unique concept. It's been 14 years. I came there in 2008 from Oklahoma. I was with USDA in Oklahoma. Moved there, and one of the last people there, right? We've had a lot of changeovers over time. But the whole idea is that we have at least one PI from 8 universities in the North Carolina system, including Duke. And bringing everything from clinical trials to epidemiology, to basic lab science, even agriculture, which is where most of my focus is, to come across and change people's diets, change their habits, and look at chronic health and see if we can solve some of those problems. The Plants for Human Health building is 4 stories. People tend to think of us as a center or an extension location, and we're not. We're not a pole barn. We have brick buildings. We're beautiful. We have beautiful labs. It's a great place to work there. Never have a problem attracting students because it's such a nice, peaceful, beautiful place. So we have a number of, just in the Plants for Human Health Institute, we cover a number of areas. Horticulture, probably our biggest group is food science and bioprocessing. And then we also have people that are in the food chemistry, animal science areas, biochemistry, plant biology. So we try and cover a number of things. We have 13 faculty just in our building. Across the campus, we have 47 faculty from different universities. We're up to, I think, about 200 million now in grant funding across the campus. All right, so back to elderberries. So my plug is done. So what's an elderberry? Whoops. Okay, that was interesting. Okay, so elderberries have a lot of phytochemicals, and the whole point is to try and take advantage of this, as we heard earlier, take advantage of these to get value-added products. How do we go from these berries, tiny little berries in a cluster, to a product that everybody can pick up and use every day? And then the idea is to use these things and expand our markets a lot, get a little bit bigger from where we were. We know that there's anthocyanins, mostly the cyanidin group, as we saw earlier. A lot of the flavonols, mostly quercetin in this group. And then the other thing that affects the bioprocessing side of it, the food science, is that they're relatively high in sugars. 8 to 13% soluble solids. We're talking similar to a blackberry. So this becomes an issue when you're doing food processing work. And also the ascorbic acid and vitamin C, or it's same thing, in elderberries can range anywhere from 6 to 25 milligrams per 100 grams. This can be a good thing or a bad thing, depending on whether it affects pro-oxidant or antioxidant as you're doing the processing. Okay, so this is another one, trying to accumulate what we know for products right now from elderberry. We know that the syrup is by far the big winner right now, right? Especially COVID-19 has made it the big winner. There's fruit that is sold, but it's primarily going to be syrup or juice or concentrate. The powders are a relatively new thing. And the point of the powders is that you can start incorporating that into other products, especially additive ingredients. You want to make a product, maybe it's for the supplement industry or a colorant or something like that. In that case, a powder is going to be the easiest form to transport and store. Now, the problem we run into this is where Karuba and Roberta have stepped in, because this is where my food science stops. Anthocyanins can be very unstable with heat and light. And if you're trying to make it spray-dried products, this becomes a very critical question. And again, the high sugars, they make it very hygroscopic or sticky. So you get your spray-dried powder and it immediately kicks up on you. How do you stop that from happening? Once the water gets in there, you're going to accelerate certain oxidative properties as well. And then the ascorbic acid may protect or may not. It may degrade the anthocyanins. So how do we extract it? How do we get the fruit from the seed, make the juice of the pomace? What formulation do we use? Dry or freeze dry the fruit, liquid or powder? Again, here we're going to be talking more about the spray dries, and we'll explain a little bit why. All these decisions have to be made. What type of temperature, what type of time does it take to do this? And then, of course, we haven't even got here yet. How do we store these? What temperature, humidities, atmosphere, probably no light. That's almost a given. And how do you handle these? How are we going to, obviously for us, we're interested in the powder end, but if you are somebody who's interested in concentrates or frozen liquids, how would you handle these and how would you store them? And then when you go back to the powders here, there are carrier agents added as well. And Roberta and Karuba will cover some of this. Now, why are we using spray dried powders? Mostly because of the many alternatives we have. Freeze drying would be the thing that's closest to quality in a spray dried product, but it's extremely expensive to do and quite slow in terms of transport and use. Right now, what we're doing is basically going to develop the powders. So we want to take this to the next step, which is to look at the studies for brain health and animal work that needs to be done with the product. And then eventually get enough, well, hopefully not eventually, quickly get enough information so we can figure out how to formulate the value-added products for the general market. Okay, and then I'm going to pass this over. Still mine? Oh, all right. We're so rehearsed. Okay, so this is, again, the overview, the schematic overview. You start with your elderberry fruit, and then you produce your juice, your concentrated extract. Well, one of the other things that we didn't really see coming until Karuba was doing it was pomace. What do you do with the pomace? There's a lot of seeds in there, a lot of bits in there. Could that be useful on its own as well? And boy, there's big piles of this stuff. Some people usually recycle it for animal feed. But of course, there's always that tickle in the back of your mind, can this be another source? Especially oils, possibly, from the seeds. So then the next step is, once you have this concentrate, you have to figure out what your matrix is going to be. And then, okay, sorry. Yeah, and then do the bioactive composition and bioavailability. Spray drying, you're going over, let me find my little, here we go. All right, spray drying is going over here to your powder. Select your ingredient, scale up. This is a big step for us, by the way. We have a pilot scale processing plant. It's called NC Phil at our center, which makes, and it's devoted to plants, by the way, because we have a dairy and meat one across the state as well. So that makes scaling up a whole lot easier because it's on site. And Karuba will be sending a student to do just that, come out there and work on it. Formulation and production of the elderberry-derived model food. And this is the long scope here, because eventually you have to do sensory evaluation. That's a few steps away. And then also looking over here, this will be Dr. Greenleaf's bit here. looking at bioassays and start following their uses for brain health and other health issues. Okay. All right, now we break.
Okay. So here is just, hi everyone, my name is Roberta Hoskin, and I will continue what Penny started, but what, higher? Okay. So before I get into what we envision in our plans and everything, let me introduce myself, because I'm probably the newest kid on this block here. So I just want to show, like, briefly where I come from and what's the link between my past and now. So I'm a chemical engineer, and I have a master's in nutrition and a PhD in chemical engineer. So I'm kind of this hybrid person that navigates between the chemical engineer and nutrition side, passing through chemistry and food science and all these things. So for 20 years, I was a professor in a Brazilian university called UFRN. It's a decently large university. We have more than 40,000 students, 2,000 professors, and more than 200 graduate programs. And during that time of my life, I was serving at the chemical engineering department. I was doing a lot of teaching and research. And mainly all my teaching and my research was mainly dedicated to food engineering students, but also chemical engineering students. And then in 2015, I came to, actually my relationship with the US is longer than this, because in 2003, I did part of my PhD in Massachusetts. So I've been coming back and forth for a long, long time. But in 2015, I did my sabbatical at the Plants for Human Health. That's when I met Penny, when I met Marianne, and all the other people. And then after I left in 2016, we continued our partnership. And then now, since 2021, I'm a senior research scholar there. And I work both at Marvin Moncada's lab that is here today, and Marianne Lila that could not come. So also another thing that I would like to highlight that has some relationship with what we are doing right now is that I come from Brazil, and I come from the Northeast, and the equator is right here, right? So this is all Northeast. So it's very tropical, and all my life, like during these 20 years, one of my main interests were the investigation of bioactive potential of underexploited or undervalued fruits and pomaces, because we also have a huge amount of pomaces, and many times the producers don't have no idea what to do with those warehouses full of stuff, right? Another thing that I was always very interested in was to develop novel ingredients and with preserved phytochemicals and also enhanced functionality. And also the assessment of the impact of all this processing on the phytochemical compounds of these fruits and vegetables. So I think that this says a lot why we are here today, why I'm here, why Marvin is here, and because I think that I see lots of these things that were in my past also in this elderberry project. So like you guys already mentioned, Penny also mentioned, so we know that elderberry fruits have a lot of phytochemicals, right? But we are also very interested in the elderberry pomace, because at the Plants for Human Health, during all this time, we have been working a lot with pomaces, not elderberry only, of course. We have worked with cranberry, blueberry, cocoa, blackcurrant, like you name it, lots and lots of berries, because the guys from the industry, they want to take maximum profit of what they have in-house, of course. And they want to find creative solutions for those pomaces, those secondary streams that in theory, they are waste products, but in fact, they still contain lots of phytochemicals that really interest us. In fact, this manuscript here said that in the elderberry pomace, we have around 80% of the originally tannins found in fresh elderberry. So contrary to elderberry fruits that have been a lot investigated, there is a lot to do, still a lot to do regarding the elderberry pomace. And also something that I would like to talk a little bit today would be about drying, right? Because in our section, in our food science group, our goal in this project, in this grant, is to develop powders from elderberries that have value added to it. So many times when we talk about dried fruits and vegetables, we think of something like this. But in fact, there are ways to produce powders, beautiful powders, with a way more versatile format than dried fruits like this. So different drying techniques lead us to a completely different product. So this very traditional way of drying fruits and vegetables usually are obtained through hot air drying or tray drying, or even sun drying, which is the most traditional drying technique. But in this grant, we are interested in using spray drying to produce the elderberry powders. And one thing that is very different from this type of drying and this one here is that spray drying is an atomization process. So we force our feed mixture to go through this nozzle. And that's the reason why the spray dried powders have this completely different texture, and they're very smooth usually, right, when we are successful. So why spray drying? Actually, let me say that I'm not an advocate for spray drying. I love spray drying like I love every drying technique. I have worked with many, many, many different drying techniques. But in fact, the market calls for it, like the industry calls for it. Many times, like Marvin is here and he will not let me lie, right? That many times we kind of offer the industry, we have other options, but they want spray drying because it's very convenient. There is a market for it. And also it's very easy to scale up. Like Penny mentioned, one of our goals in this grant is to scale up the production of these elderberry powders. So through spray drying, we can obtain lots of diversified products. And compared to other drying techniques like freeze drying, it's way more economical, right? So these are some of the reasons why there is a lot of interest around spray drying. So if I have to summarize, like what is spray drying, I would say it's a very quick method to transform a liquid solution into a powder. But in fact, we can use, it's a little weird right there, but actually we use, it's very flexible. We can use fruits, extracts, fruit juices, you name it, but mixed with proteins or polysaccharides or in other carriers, and we pump this feed. Oops, I'm sorry. We pump this feed into this chamber at the same time that we pump hot air. And we can control the temperature of this hot air right here. So many people have this question, wow, spray drying will destroy my bioactive compounds because it's hot air getting in contact with the liquid that has the phytochemicals. I always say every processing induces damage. You know, it's not, we are not naive enough to say that processing is perfect. But in the case of spray drying, this is an instant evaporation. The time of the contact of the hot air with the liquid solution is like instant, like seconds, milliseconds. And we have some controls around these temperatures and the inlet temperature, the outlet temperature, blah, 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 right? So I will not get in super in depth of this, but believe me, it's true. So the feed solution gets inside this chamber and the powder stream is forced into a pathway. And in the end, we collect in a vessel. So in few words, this is spray drying. So three things that are very desirable around about spray dried powders is that, of course, like every powder or dried food, it has an extended shelf life. But another thing is that spray dried powders are very smooth in general. So there are many other drying techniques, for example, drum drying, or even freeze drying, or refractoriness window drying. In the end, you end up with flakes, not with powder. So you have to add one more unit operation to your process to grind, to get to that texture. So spray dried in general, we can get, our final product is a very beautiful, smooth powder. Actually, Kiruba brought some samples here and she will show it to you. And also, another big reason is the versatility of the spray dried powders. Also, because they're easier to storage and they have extended shelf life, we can think about many ways to use these powders to create different products. Just an example, like recent projects that we have done, we using a microwave assisted extraction and processing, we have produced some smoothies made with spray dried powders. We have worked to a big company that I cannot say the name, but we end up with these powders that they are very interested in adding it to their cereal bars and their soups and lots of different applications, not only fruits actually, because we also work with herbs. Like we have done lots of interesting things with spinach, with rosemary. So it's just, I just wanted to give a little taste of our vision and why we picked spray drying for this grain. So I will pass my friend Kiruba, who will finish.
Thank you. Thank you, Penny and Roberta. And it's been a wonderful project. So yeah, now, since Penny explained to you about the importance of elderberries and Roberta explained to you about the importance of spray drying, I'm going to go in deep and discuss about some of the results here. And before going, I would like to introduce, I'm Kiruba Krishnaswamy. I'm an assistant professor at the University of Missouri. And before coming to Missouri, I'm from India. So I went to TNAU, that's a land grant institution, one of the oldest land grant institutions in the country. And then from there, I went to McGill to do my bioengineering, and then University of Toronto chemical engineering. So we all share a similar hybrid of food and engineering here. And so some of the requirements, when we do spray drying, some questions we ask is, how do you determine the viscosity of the materials? What are the ideal temperatures that we can spray dry so that our pigment, the powders that we get are not reduced in their quality? And what type of carrier matrix we need to use? So we got some frozen samples. In order to do our studies, we wanted to do some preliminary testing. So Andy was kind enough to give us some old frozen elderberries, and these are the cultivars that we got. So apologies, there's a video here. It's not playing today, but tomorrow we'll make sure it plays. So we use the frozen elderberries to pulp them using the RoboCube, and then 1 mm mesh size, so we can separate the pomace, the seeds from the pulp. And then it was centrifuged and to get the juice separately. So as Roberta was saying, there are a lot of parameters that goes in optimizing the spray drying conditions. So first thing we need to understand is what temperatures are important when we spray dry, so that does not degrade the bioactive compounds. So this is a spray, a spray dryer, this is the juice that was meant, and these are the different temperatures we tried, inlet temperatures and the corresponding outlet temperatures. So you can see, like with different temperatures, you can see that, so with different temperatures, you can see the color changes, right? It's not quite clear here, but we have an equipment, the colorimeter, which can give us the exact values of L, A, and B. And when we calculate the total color change, like keeping the elderberry juice, the fresh elderberry juice as the reference and the spray dried materials as our samples, then you can see for the color change, this one. So the 100 degrees, the spray drying at 100 degrees, 120 and 140, those were like similar to the materials that was close to the fresh juice. So when we did the screening, we got to know, okay, these three temperatures kind of gives us a better samples. And also when we have some physical observation, the ones with 100, there was a lot of medium electrostatic. So the powders will stick to the walls of the glassware. And then with 120, it was a very free flowing powder with less electrostatic force. So, and then with the 140, it had high electrostatic properties. So when you have a lot of electrostatic, imagine in an industry, when your powders are going to get stuck in the walls of the spray dryer, so your yield is going to reduce, right? So we want to have the maximum yield with minimum damage to the bioactives. So that led us to select 120 as our next step. So how do you measure them quantitatively, right? So we need to have color values, colorimetry, and we can calculate the Browning index, and that's like the physical characterization, and the chemical characterization, the anthocyanins, ascorbic acids, and so on. So we did a series of physical characterization to understand the quality of the powders that we were generating. So we calculate the color values using a colorimeter, total color change, hue angle, chroma, Browning index, yields, moisture content using the moisture analyzer, and bulk density porosity using a pyconometer. So we did a lot of experiments. I'm just summarizing the key optimal conditions that worked well. Conditions were 100 degrees, 120 degrees inlet temperature with 100% aspiration and 10% pump. This is because it comes back to Penny's discussion on the bricks. The sugar content of the elderberry is quite high. So so that we have to introduce the sample in a slow rate so it doesn't stick to the wall materials and you can get free flowing powder. So we were able to find that. And then we wanted to try with carrier matrix. What we did is we took elderberry and pulped it to get the filtered juice, and then we mixed it with soy protein isolate and tapioca starch, and that the same thing was introduced into spray drying. So this powder is elderberry juice without any carrier matrix. It's pure elderberry juice. We were able to spray dry it. And then this is with tapioca starch, and this is with soy protein isolate. I'll show them later. It will be kept in display. So if you see them, like, there is a little bit of color change for different combinations, and we have different carrier matrix. So the tapioca one was very hygroscopic in nature, and there was visible agglomeration. And the soy protein one had less hygroscopicity as well as less agglomeration. And so we did a color analysis, and what we found is, like when we was tried, this is something that I want you to see. The B value, that is the soy protein isolate, that kind of increased the B value, that is the blueness of the powder. And all the other things, the L values, there was significant difference there. Like L and A, they all were pretty much, but this B value, that was something very interesting for us. And you can vividly see it in the powder as well. And the Browning index, like from the total color change and the Browning index, what we can see is the total color change was higher for powders that was produced with the soy protein isolate, and the Browning index, this shows the least degradation. If you have less brown color, that kind of signifies that there is less degradation that was due to Maillard reaction. It's a reaction that happens between amino acids and sugars. So that is a strong indicator saying like soy protein isolate could be a better carrier for juice. And then we used moisture content and also the water activity. Water activity is pretty low, so that's good from a microbial standpoint. And there was an increase in the moisture content here that might be due to the holding capacity, water holding capacity of the proteins. So that gives us an overview here as well. And then with the picrometer, we'll be able to calculate the densities, the bulk density, porosity, tab density. And these values are very important from a processing standpoint if you want to store your powders, like, in a big containers. So here you can see the bulk density for soy protein isolate was also quite high. And with those density values, you can calculate the flowability of the powders using Carr index and Husner ratio. So here what we were able to find is the soy protein isolate had the lower Carr index and also the Husner ratio, which means that this powder is more free flowing, okay, and less agglomeration and less electrostatic interaction with the walls as well. And then we did some analysis to see what is the microstructural properties. How does it look under the microscope? So all these things are like scaled to, this is the 10 micron scale. The problem with the elderberry powder without any carrier matrix is it just is very hygroscopic. So as soon as you open it, it's so hygroscopic. So it will be, from a processing standpoint, it'll be difficult to scale it up. And you can see, as soon as you put this thing on the stub, it started absorbing moisture. That's why it's a lot irregular in shape, all these materials. Then if this is the image for the elderberry juice with tapioca powder. And here also you can see some dents and wrinkles on the surface. And also this kind of correlates with our browning index, where we had a lot of browning for the tapioca. Could be due to the reactions, Maillard reactions. And then this is the elderberry juice with the soy protein isolate. And here we could see more spherical structures. And also, like, there was a few agglomeration that could be because when we are opening, there's some moisture interactions there. But comparative to other samples, this kind of performed well. So here's our timeline. The year one, we had the production of elderberry ingredients, like we were trying to see what combinations, what processing conditions performed well, and to optimize them and kind of have that completed. And now we're going to evaluate, do a comprehensive study on the chemical properties as well to compare with the juice and the pomace, and then that will lead to scaling up at NC State. So we'll do our screening both at MU and NC, and then we'll get the best formulation to going for scaling up. So that particular powder can go for the brain study. Thank you.
So we're going to take this another step further. Dr. Michael Greenleaf from University of Missouri is going to give a fairly brief overview of the brain health research, which is starting soon. Dr. Greenleaf, thank you.
All right, I think, so then what I'm going to talk about are some proposed studies. These are studies that we're going to actually be starting in the next year, or next year, the grant, but this fall. And it's going to build on some of the information that both Chung Ho and Karuba just talked about. And so I'm Mike Greenleaf. I'm in the Department of Chemistry. I also run the Proteomics Center on campus. And so what I'm going to be talking about is what we're turn this around. What we're going to be doing with brain health. Okay, so we've heard some about what's in elderberry and the natural products, and some of the antioxidant behavior. Also some about food science just now, and kind of the last part of this is trying to tie these parts together into some brain health studies. Okay, and so the members of our team are Zhejiang Gu and Janice Ku. They're in pathology and anatomical science at the MU Medical School. David Beversdorf is in radiology. Ashley Curtis is in psychiatry. Grace Sun is a retired faculty member from biochemistry who's also going to be helping. And then one person I accidentally left off was Jack Chen, who's our bioinformatician and computer scientist, who's going to help with some of the data analysis when we get to that point. Okay, so I'm going to kind of divide this up into kind of like what we know and what we're planning to do. So what we know is that quercetin is a flavonoid. It's commonly found in a whole variety of different elderberries. It's a representative of a polyphenol. And we've done several studies in microglia cells where we know that it helps reduce inflammation in those cells. So we can microglia cells, a common cell that you find in the brain. If you stimulate that to have it generate reactive oxygen species, so the cells are under stress, that if we add quercetin to those cells, the stress level goes down, okay? And so here's just a number of studies we've done over the years looking at that. Okay, if you like more detailed biochemical parts, this is a diagram dealing with that. So what we use as a stimulant is an endotoxin called LPS to stimulate those cells. And in nature, well, in the normal state for microglia cells, if you add LPS, that tends to have it will generate reactive oxygen species as part of that stress. It also has arachidonic acid. That's what ARA stands for. And as the arachidonic acid is transformed in a process called lipid peroxidation to make these small molecules right here that are alkenols here. Okay, if we add DHA, so DHA is a fatty acid that's commonly found in fish oils, like salmon oil. If we add DHA as a fatty acid, and if it's oxidized, it produces a different molecule called 4-HHE. And what we found with DHA added to microglia cells is it will also help reduce stress. Okay, and so what you see is an increase in HHA being produced and a decrease in 4-HNE. Okay, and that just it's the way that those cells are accommodating or alleviating that stress that you've induced in those. Okay, so we know we see that effect with quercetin. We know that we see that effect with DHA. What happens if you put the two together? Okay, and so this is still in microglia cells. If we put the two together, the main point is right up here, is they work better together than individually. Okay, and so what you can see is this graph, or this part of this histogram, this shows you the amount of reactive oxygen species that are produced when you add LPS to that to start generating those reactive oxygen species. As you add quercetin and DHA together or by themselves, that will go down. But combined, it goes down better than each of them individually. Okay. We can also follow that with those small molecules, the 4-HHE and 4-HNE, and those follow. Go back. Okay, and they follow the same kind of trend. Okay, so they overlay one another in that respect. Okay, so now we know that they work better together. The other thing that we can do, and I'm not going to try to go through all of this part, but we can also look at what's going on in the cells at the protein level. Okay, so I take these microglia cells and I use a method called proteomics. So in proteomics, what we're doing is we're using mass spectrometry to identify as many possible proteins that are present in the system, as well as their concentrations. Okay, so and then this is a heat map, or what's referred to as a heat map of the results of reacting DHA with these microglia cells. Okay, so going across the bottom, these are this is DHA here. These are 3 control samples. If I had LPS and LPS plus DHA. And the 1, 2, 3 just means that we did this experiment in triplicate. So to check the reproducibility of that. Okay, so the one thing you can see right away is you can see regions of the map right here where you have LPS added and the concentration of those proteins go up. And when you add LPS, with DHA, that goes down. Okay, and then over here, those are actually the names of the proteins. But most of the proteins that you see going down are ones that are dealing with relieving antioxidant or stress in those cells. Okay. So that's what we know kind of the background studies. Okay, the other thing we can do with that we've been able to do with DHA is we can extend this, we've extended this into a mouse model, and we've been able to show that we can follow changes in different body parts of these mice as a function of time. So if you feed the mouse, in this case, we were feeding mothers DHA, and then we were detecting whether or not that was being passed on to offspring mice. Okay, and so if you look in their heart, for example, you can see the amount of HHE goes up quite a bit. Same thing on plasma. So these are molecules that you can easily, point of this is these are molecules that you can actually measure. The harder part was, can we see those kinds of changes in the brain? Okay, so this is a post-mortem study where we look at different parts of the mouse brain. So this is done with microdissection, where we can look at the cerebral cortex or the striatum and look at changes of those same small molecules there. So we have a way with mass spectrometry of being able to follow these changes is in the brain. Okay, so that's kind of what we know to date, kind of to set the stage. What we plan to do, and where we plan to start this in the fall, is we plan to work with an aging mouse model to try to see whether or not giving mice elderberry will affect their mental cognition. Okay, or how that may or may not affect their mental cognition. Okay, and part of the reason for doing that is in the hallway, there's a poster. I'll plug the poster that Ashley Curtis was the lead author on, in dealing with a small trial of giving elderberry juice to some people that have some mild cognitive disorders, and comparing that to a group of patients that did not have that. And what we see is that there's some improvement in their mental cognitive state over a period of time by giving them elderberry juice. And they were taking, I think it was, and Dave, if you're in the back, you can correct me if I'm wrong, was 5 meals a day for up to 6 months. Okay, so there seems to be an effect in people, and we wanted to first see if we can say what's going on in a mouse model. And so the way we're wanting to construct this study is we'll have a control group that are just going to be mice that are aging mice that are taking a standard diet. We'll have another group where their diet has some DHA in it. That'll probably be about 1 to 2% DHA. We'll have one group that'll be given at some elderberry powder. That'll be courtesy of the food science group using the same powder that they're developing as part of that project. And then we'll have a fourth group that has the two of them kind of tied together. Okay, and so this is trying to build on what we already know with quercetin and DHA added together in the microglia cells. And then what we want to do is we want to monitor levels of these two small molecules in serum as a function of time. We can also work with Lloyd Sumner, who's in the Metabolomics Center, and he can also look at the small molecules that are present in the serum as a function of time. And at the end, we'll also take a look at the protein level changes that are going on, and we'll also take probably also do the metabolomics of that. So a couple definition terms for the non-scientists or the scientists who might not know it. So proteomics is trying to understand all the proteins that are present in what you're studying, and using that and identifying what those proteins are by mass spectrometry. Metabolomics is the same kind of technology, but instead of focusing on proteins, you're focusing on small molecules. So small molecules typically have masses of less than 1,000, and some would even argue smaller than that. Proteins tend to have molecular weights or masses above 10,000. Okay, so different size systems, and but we hope to be at the end, be able to combine both of those kinds of studies together. And then that's where Jack Chin is going to come in to try to tie together the metabolomics and the proteomics data that we'll be generating. Okay. Another thing that we're going to try is to understand a bit about the mechanism that's going on. So it's one thing to identify, oh, I have a certain protein. It's increased in concentration by giving this mouse elderberry. But what does that mean? So that has to do with what's the pathway with which is that's occurring within the body, okay, or within the mouse body. And so one thing we can do is we can use what's called a signaling pathway reporter. And we plan to work some with Val Mosina about this, who's a biochemist at the University of Missouri. And he has a series of reporters that are divided out by a whole, by different kinds of flavonoids, or I should say by different kinds of phytochemicals. And then they are specific to different kinds of pathways within mammalian systems. So for example, NF-kappa B is one that's really tied to brain health and process that deal with inflammation in the brain. Okay, and so by looking at these different cultivars of elderberry, along with these reporters, we can get an idea of where changes can be occurring, and then can we correlate that with the proteins that we see changing also. So it's not only saying what the protein is, but really what's the function of it and how that may play a role.
Wow, very interesting. So we're ahead of schedule, still very good. We're going to shift gears kind of from amazing science to maybe more applied. So we're going to have a shared session here with our nonprofit partners, the Kerr Center for Sustainable Agriculture in Poda, Oklahoma, and the Savannah Institute. Where are you guys based? In Madison? I'm not sure where your home is, but so I think David Redditch first, about 10 minutes, and then Fred, forgive me, Fred Utsi from the Savannah Institute. Just kind of explain a little bit about who they are and kind of what we're doing, and kind of back to horticulture. David Redditch, take it away. You guys have a slideshow?
Yeah, is this it? Okay. The down button? Okay. My name is David Redditch, Kerr Center for Sustainable Agriculture. We're located about 30 miles southwest of Fort Smith, Arkansas, to give you a geography of where we're at. We've done some work with elderberries. So we did a specialty crop block grant in 2018, 19, and 20 through Oklahoma Department of Ag, looking at 5 different varieties of elderberries. Some of the growers in Oklahoma that are doing 360 Farms and Shortline Elderberry Farm are both doing work, growing elderberries, selling potted plants and selling elderberry products. Brett focuses on, and his wife focus on elder flowers for teas, and they've also moved into wines. Shortline does potted plants, and they do some elderberry products at farmers markets. There's a lot of elderberry growers in Oklahoma doing the small scale production systems. And the research, as far as I know, we're the only ones doing any work with elderberries. We did 2 different plantings for this study in 15 and 16. And those are the varieties, but they call it, the soils it's on is called Worcester silt loam. I'm originally from Missouri. I grew up in eastern Missouri. The definition in Oklahoma, what a silt loam is not the definition of a silt loam in Missouri. They're very different soils. Anyhow, I looked at bricks, yields, berry weight, and then the plots were not harm pruned until the first year of the project. So that's what the project looks like. And there's Simon, he's back there. He's running the weed eater to cut the plants down. And then we haul them off. Now, one thing then we're kind of fertilizing with, that's feather meal, slow release nitrogen on there. But this is what we're running into, some very heavy rainfall events, rapid, and the water will drain off, but we've done some modification of the plots to reduce this. But then this year, we're setting up for this trial, and this is what we experienced. And I mean, the levels, the amount of rainfall we're getting in quick, this was about a 20-minute rainstorm, and it dumped on us. And so, and those are where the plots are at, so are going, they haven't been planted yet. So this is kind of some of the things we're experiencing with, I don't know if you call it climate change, whatever you want to call it, we're experiencing very heavy rainfall events. And the challenges we're seeming to come up with is shattering before ripening and during harvest, uneven ripening on our berries. Weeds have started to become a problem for us. Up until the second year of the study in 2019, weeds were really a non-issue. The elderberries seemed to be vigorous enough to keep them down underneath the plants. And I will say, do not let hairy vetch get started in your plantings because it binds up and pulls the plants down. And I imagine it would hinder any type of commercial harvesting. Secondary blossoms seem to be coming up after the first ones come up. I don't know if that's due to stress resulting in a limited second harvest. Mite damage. The final year of the study in 2020, the mite damage was so heavy on the planting because I was not trying to actively control it. I literally had a crop loss over all the studies, and it was bad enough that and this is the publication, it's online, and I have some copies here if anybody's interested. But the last year of the study, I kept the results separate because I didn't feel they were reflective of what could happen if you actively tried to control the mites. Heavy rainfalls, I'm concerned about nutrient leaching. So one thing I like about this study is we're going to be doing some leaf analysis to determine the level of nutrients and find out what's going on. Elderberry borer. This year, I'm going to show you a picture in a little bit, and birds. I don't know how bad they're going to be. There's the uneven ripening. There's some of the mite damage. This shot I took with my cell phone, May 25th. You can't tell in here, but there are 1, 2, 3, where's the other one? 4. There's 5 breeding pairs of elderberry borers in that shot. And when I walked up and down the rows, it was an overcast day, you could see elderberry borers everywhere on 2 rows, 200-foot long rows. So I don't know what kind of effect that's going to have, and why they showed up this year, but it was an overcast day. And then this shows you some of the weed issues with the fescue in there, but and this is the beginning ending harvest dates on the varieties that were used in the trials. I've got it in this, you could read it better in the tables in this study, and this is some of the yield levels that we were seeing. And the Brixes are right in line with, hold on, back up. The Brixes are right in line with what we were talking about earlier, about 13 to 9%. The Brix levels held pretty high. One thing this did not take into account is the number of unripened berries in there. So these were total yields. I would have to sort out the unripened berries, and that would drop some of those yield levels. And this is the trial planting that's in the ground right now with the irrigation laid in. I do have some Pocahontas I bought from Terry last year, some plugs. I'm babying them in this raised bed so I can take cuttings off of them. But those, I'm 5'10", those blossoms are above my head. They're up here. So, and I did one thing in the study I did, it was trying to take plum size measurements because Brett was doing teas with the blossoms. One of the issues I had with it, they're reported in the study, but the problem I had was the blossom densities varied, and there was no way to factor that in. So I questioned how accurate taking blossom measurements are. And also, if I was going to study the value of blossom teas and the effect on an elderberry planting, that needs to be a separate study from berries, because how vigorous will the plants be after the blossoms are cut off and the plants are not allowed to go to full berry? You may get a much vigorous stand going into the next year. I don't know. So
we have one more presentation. Dr. Zhu is here, Dr. Zhenfang Zhu.
Good afternoon. I guess last group, I don't want to take a whole lot of time, and probably all the tired about the science. So we go like something engineering, okay? So we'll talk about a mechanical solution, not only for harvesting, we also talk a little bit on the post-processing part. So we have a couple of this team, like it's me, Jianfeng Zhou, assistant professor in University of Missouri, And we have Leon Schumacher already left. So Krupa also on the team, and Lang Hei is in Penn State, cannot join. So have several students and yo.
Hi guys, I'm Joe Barada. I'm a mechanical designer for all of this that I've worked in ag engineering for a while now, and I came up with actually the system of how we process and do the post-process cleaning for elderberry.
Yeah, we'll show that. Joe will talk more about those.
And I'm Gianluca. I'm the electrical and computer engineering in this group. I'll be working on the camera system as well as in the robotic implementation of what we're going to present.
Yeah, I have another student not able to join us, Rifat. He's like mechanical engineer background. So basically this is our team. I'm going to focus on develop something. So a little bit background about down here. Okay. So okay, so I have some experience work on sweet cherry and apple harvesting. Before I joined Mizzou, I was in Washington State. So we developed different harvesting system for fresh market, cherries. That's very challenging too. So we want to review a little bit. We don't have too much have been done this year due to this. We're missing the season last year. Because you develop harvesting system, you want to understand the orchard, the tree architecture. So we don't have too much, chance to go to the farm last year. So basically, we don't have too many ongoing activity. Plus, my student came late, so we just want to give an overview of our thoughts and how we're going to move this project forward. So just a little bit about the goal. So we really want provide some efficiency so you can scale up your production. In terms of our project, we want to focus on in orchard what you do, right? So remove fruits, use a harvesting, mechanical or robotic, whatever solution that's feasible. And then we also want to see how you can collect those fruits, remove from stem. So that's about the handling part. So we tag this two together. So there's some of our plan. The first year we want to really evaluate the existing technology, either theoretically or if we have a chance testing in farm, we'll do that too. And in next year, hopefully we can develop some protocols that can test in field. And also want to have some activity for the fruit quality assessment. We're going to talk about that later. So I will jump up this one. So basically, if you look at the current existing harvesting technology for berries, as I summarize a little bit, basically just using the shake and catch. A lot of, if you know some of those berries, you can see this is a very typical structure how they remove fruits right now. Either vertical vibration to shake the fruit off from the tree or the branches, or use this, it just fit to different architecture of your tree. And some of these days, if you know the one grape, that's how they harvest. They sweep the canopy or they beat the canopy, that's how they remove fruit. So in several of the last slides, I just want to show some example. We find online, there's some, if you look here, that's how they structure it, right? This is probably just collecting fruits and use a conveyor to transfer fruit to the collector. But, in terms of harvesting technology, that's majorly, you see this one, this is shaking the canopy. What you need to do is to optimize the frequency, amplitude, the motion you need to do. So this is all very similar. This is another example, this is the same way to remove it. And here is, it's just different example. The actuators a little bit different, how they structure it. For example, this one is not only shaking this way, it can rotate. It's just trying to moving the canopy and find the fruits. All kinds of optimization, but in general, their method is just shake the whole canopy. More example, right? This is just all different, fit in different trays. Like there's the existing technology. When we think about that, what about the elderberry? Can we use this or not? This is really a good question. Something we need, okay, this is another example too. I mean, there's a lot of similar products available for different small, fruits. So this is a non-selection harvesting. Are you happy with that or not? When you shake your whole elderberry, you probably will remove both mature fruits and the green fruits, right? Okay, so I hope this is hard to address unless you have a good variety. That's what depends on the breeders. If you have a good variety or some agents make them mature at the same time, this will be very useful. The second, there are a lot of damage could happen if there's mature fruits. Again, based on my experience with sweet cherry, when we shake the tree, a lot of roof damage. It depends on energy. If you the energy is not enough, it's hard to remove the fruits. If you're shaking too much, there will a lot of damage. So this is hard to address this kind of question. And what about the property of the push, right? The difference with the tree. When we use sweet cherry, this is a tree. This is a kind of more soft architecture. It's bring another challenge to remove the fruits. And also, if you think about the fully mechanical solution, what about the cost? What do you think one system like that, how much it will cost? How many farmers may affordable to this type of machine? So there's a lot of parameters we need to think about when we talk about the fully mechanical, non-select or mass harvesting solution. Okay, so what about other solution? We also think about there is I call the mechanical assistant. It's not a fully mechanical solution, but what about use some machine to improve efficiency? For example, this is the apple. This is just a platform. Pickers will just standing on this machine when you save the time travel back and forth, and you can reach different height. Is that could be a solution or not? We need to discuss with the, growers and some others. Another part, like, do you like this? So you can use a human to assess the fruit, which part I want to harvest. Then you have this. This can be a, is a pump, hydraulic or pneumatic, whatever shaker that you can shake a certain part. That will improve your efficiency. That's possible or not. So even more like really just use this, right? So all there's kind of solution making thinker. That's what we're going to evaluate in the following several years. All kinds of mechanical solution, which one can potentially improve, the efficiency, make everybody, not scared of this labor shortage problem. Anyway, that's just some thoughts we needed to talk in the following years. So another possible, okay, what about robot? You see there's robotic solutions for a lot of high value fruits production, apples, cherries. So these two examples, those are what? Those are strawberries. So this one in Florida, you can see a lot of this, not a lot. I mean, there's a company work on this, is demonstrated working well. There's another way, right? There's a robot arm, is down here. They have a camera system. There's another robot arm, they just, pick up the fruits. So those possible or not? I mean, so we don't know, since the challenge for this one, they have so many fruits. What about our elderberry? It's in the head, in the cluster, will be a better solution or not? So we're going to evaluate, and that's some idea we have, right? Like I said, we don't have a whole lot of stuff have been done yet, but in the following years, since my student team already here, so we're going to try some different technologies. One of the things we want to start with, maybe we're going to evaluate is that robotic solution possible or not. I didn't find a good harvester yet. Andy know that it's very expensive to buy one. So what about we just start some simple solution right now? So if you look here, we have different cameras, 2 cameras here looking both sides. So we detect the fruits and we build the 3D model, use AI technology. Even we identified, the ripened fruits rather than, you remove all the fruits. So we have a robot arm to pick up, just cut the fruits. So that's some solution we want to try. And so this guy is a computer and electronic engineering. He's trying some camera system. Go ahead.
All right, so I've been working with a depth camera, which uses stereo vision or 3D vision to model an image into a 3D point cloud or world, right? And we use this in order to detect the position of, our goal is to detect the position of the elderberry cluster. I use this example with some clusters of lavender plants, and we use object detection algorithm to able to identify the clusters. So by localizing each cluster, we can interact, get our robot arm and interact with the cluster in provide a path planning algorithm in order to grab it and cut it and deposit it into the depository bin. So that is our goal that we're working on it right now. Currently, we are thinking about 2 depth cameras in order to get a better view point and also with less noise in the images. Yep.
So for the depth camera, it can build a 3D model. Depth means it can detect the distance as well as you can see the color. So basically to provide the color information, also the distance information for you to understand where the fruits are. So so that's we are trying a little bit, but this summer we're going to go to field to collect this type of data. We hope this at least we have some model to evaluate the fruit maturity level, and so we can construct a 3D perception map. So let the robot arm go to pick up the fruits. So then we'll work on the robot arm again, cooperating with these 2 systems. Anyway, so there's a lot of different technology already being tested. This is not new, like, for apples, strawberry, they all use the same technology. Hopefully we can have some results this year. So that's probably what we have right now. I just want to share some thoughts. In the following year, we're going to try this different technology in field. Okay, so this is the harvesting. Okay, we have a second part. I don't know how much time we have. Are we done? Okay, okay. So that's so the second part, we'll talk a little bit about the post-processing. So this is a presentation I made for another conference. Sorry for that. So I borrowed this photo from Terry's shop again. So this is we want trying to address the automation. After you getting through to the processing house, this is a Terry's shop. So the destemmer, remove the fruits from stem. Then you need to do this, right? Sanitizing, writing, and weighing, put in the bucket, 25 pounds. Then you put it in a cooler room for shipping out. So we trying to improve this efficiency. I'm going to quickly go. I'll let Jill talk a little bit. We want, this is just a simulation. So we This is the first tank. After you get the fruit here, then you have the auger shipped to another one. Then we have a, weighing system, just a digital weigh, scale. We have a controller that can continue monitoring the weight when it reach to like 25 pounds. It will stop the auger, and print a label like that. So we're going to show the system tomorrow. If you're interested, you're welcome to take a look. So I'll let Jill take over. Before that, we work with Terry. So Terry basically built another, simple solution. Use this conveyor system. So what Jill helped build this auger system. We feel like this is very interesting. So I'm going to skip this. Probably it's fine. Just a couple, maybe less than minutes. This is how the original design, Terry and John, what's his name? Yeah, built together. So, okay, so go to analysis. That's what we have in lab. We're going to bring this tomorrow. Basically it's the two augers and transmit the speed. I'll let you explain this.
Okay. So yeah, just real quick, what I was intending to do was to replace that team of 6 folks that you saw helping Terry to do all of that processing. So designed out a system to replicate those same steps and to be a single operator and to exceed the rates that they were able to put out. In terms of math, we're at about 580 kilos an hour to run it at full speed. That's with a single operator. They've got a PLC that's just going to be a start-stop, that it will automatically weigh that out. And that cuts power to the motors. And then they will take that bucket, it prints a label for you, slap that on there, put the next bucket on and hit go again. And it runs it as a single guy. He's loading the destemmer and deloading out those 25-pound buckets. Couple novel things are happening here on this, that we consume sanitization fluid as we run. So you'd have dosing pumps on that and a recirculation system. So you're basically playing with the same air gap that a plumber uses to keep solution in a set of tanks, and the berries move to the next tank without the liquid. So the solution's able to propel them along and protect it. So you don't have very much berry damage, but you still are able to bring that back into the same solution. So you can have chlorine in your first tank and then just rinsing afterwards. So you're not dealing with that and not constantly consuming just excessive amounts of cleaning chemical. On top of that, we're also using a density separation to get rid of all the field debris and the unripe berries. So skimming with a trash pump off the top, you're able to pull what you don't want and recirc out the rest. And with that kind of a split, you again minimize what's up.
Yes, you can play this video while we talking.
I have no idea what I'm doing with this.
Okay, go
I tested this with blueberries in terms of just skin toughness, and testing it with airsoft BBs, if you guys remember those little plastic pellets for actual particle size. So outside of the time that you have fresh berries available, those are our 2 decent analogs for what's going on. And you'll see briefly that the scale is that control run there. And it is a single button push for that operator. And yeah, I'll demo it tomorrow. Full disclosure, I burnt out a motor, so I'm repairing stuff tonight. But assuming that goes well, I'll have that demoed to show what's going on if anyone's interested.
Go ahead, next slide. Yeah.
Oh yeah, and here's a pretty picture of what's going on too. That if you could fathom what's happening here, that the berries are taking that path, and then there's a set of drains that allow the recirculation to keep fluid in the tanks and keep the primary tank where berries touch. So only one is ever touching fruit, everything else is just touching water and chlorine or whatever your sanitization is.
So thanks very much to our technical crew. Thanks, everybody.